Hello, everyone. Nice to see you all. My name's Adrian Marshall. If you don't know me, I'm the facilitator of the Grassy Plains Network, and we're part of the VNPA these days. This is, I think, our 37th Grassy Talk. So it's we're moving on along, which is magnificent. They're a great resource. We are very lucky today to have three students from, or ex students from the Trobe. I'll introduce them in a sec, but I'd just like to say do hang around for the meeting afterwards. They're informal affairs, but we just sort of catch up on all the news and plot the downfall of all the evil developers in the world and things like that. So that would be, yeah, it'd be nice to see you there. Um, Before I go any further, I should pay my respects to the elders past and present of the Indigenous people of I don't know, the Melbourne region in particular, so the Wadawurrung, Wurundjeri and Bunurong, but also the greater um, group of peoples who call Victoria home. And I suppose I often put in a reflection upon my own stumbling way forward to understanding these things. At GPNUs a couple of issues ago, we had something on grassy eucalypt woodland, and I was commenting that the part that's in the Melbourne Strategic Assessment's a bit sort of run down. Its understory is a bit run down. And I passed my text by one of the Wurundjeri people, and they gave me a very lukewarm response, and I didn't quite understand why that was until I realised later, well, like very only weeks ago, that it's probably because that whether it's run down or not is not really of the greatest consequence. It's country, and that's and it's important for so many reasons, and not just that simple reductionist Western sort of scientific framing of goodness of a piece of land or whatever. So that's been my little learning for the month, I suppose. So yes, great, yeah, respect to all the First Nations people of Victoria. So let's move on to the important action part of it. We're going to start with Maggie Logan. What we'll do, we're going to do each of the students will present. We'll have a sort of I'll introduce them individually, and we'll have like 5 minutes at the end of the presentation. Each will be about, I guess, 15 minutes or something, and then if we've still got time at the end, if you've still got more questions, we can just throw questions to anybody you like. And yeah, questions in the chat, but I'm sure it's you're free to interrupt our next generation of grassland researchers, and yeah, just make them jump through their performing hoops, as it were. Anyway, so Maggie Logan finished a Bachelor in Wildlife Science at Deakin back in 2022. Has been working in bushland management, and then last year started her honours research with Dr. John Morgan and Dr. Jen Wood, investigating how management legacies in grasslands have impacted the soil microbiome. So really cool stuff, and that's carried on into her master's now. And yeah, above and below ground ecosystems and the role of disturbance. So Maggie, over to you, please.
Thank you. I'll just get my screen share going. Um, I was just chuckling because when I was nervous getting ready for this, I was like, oh, it'll be okay because I'm sure Adrian will prep the crowd that like we're students and go easy on us, but not, yeah, instead it sounds like maybe we'll be jumping through a bunch of hoops or
Yeah,
yeah.
just a good audience.
Yeah, maybe not too many hoops for me, please. That's my
Yeah.
request. But yeah, all right. So my name's Maggie. I completed my honours last year at La Trobe University that I've now transferred into a master's, as you said, supervised by John Morgan and Jen Wood. And I'm just going to be telling you about the results from my honours today, which was titled The Application of a State and Transition Model to Soil Microbial Communities in an Endangered Grassland. And before I begin, I would like to acknowledge the Wadawurrung, Bunurong, and Wurundjeri people as the traditional owners of the lands where my study took place, and acknowledge their continuing connection to land, waters, and community, and pay my respects to elders past and present. So, a bit of background. A state and transition model is a conceptual framework used to categorize ecosystems within a landscape into alternate states. So these photos here are just some examples of what I mean by an alternate state. So these might all be classified, or they exist within historically what is a plains grassland landscape. But as you can see, the vegetation looks pretty different between all of these, and even whether or not they would be today be classified as plains grassland. But you can kind of see looking at these photos that they've probably had really different disturbance histories. So, state and transition models, or STMs, have mostly been developed in rangeland or semi-agricultural landscapes as a decision-making tool, but they're really useful from an ecological perspective too, because they allow us to understand landscapes like plains grassland that have had really complex and distinct disturbance histories and how that has shaped the vegetation that we see there today. So, a state is defined by characteristic vegetation structures, compositions, and functions, and transitions are what link the states, and they're built up of various drivers. So, here's an example of one that I stole from online. So, you can see that these states have distinct vegetation species, but also abundances, and that the transitions are linking certain states together. So, for example, to go from a shrub savanna state to the shrub dominated state here, that occurs through continuous heavy grazing, loss of the dominant grass type, lack of fire. And I just wanted to highlight that because the transitions, I might talk about them as quite like uniform, but they're built up of multiple drivers that work together to cause that state change. And I'm not going to talk about this in too much detail, but I just wanted to include it to highlight. This is a state and transition model that was produced for Victoria's Plains Grasslands, published by Steve Sinclair back in 2019. And yeah, I just wanted to highlight that these models have been produced for our native grasslands, and they're quite effective in allowing us to go out and understand the disturbances that have occurred within our native grasslands. So onto the microbe stuff. So a microbiome is a community of organisms that belong to the groups fungi, bacteria, archaea, and viruses, and the soil is just the ecosystem that it's existing within. So the soil microbiome is responsible for between 80 to 90% of soil's function. So that's things like breaking down organic material, sequestering carbon, and facilitating nutrients uptake by plants. So the microbiome is playing a huge role in our overall ecosystem function. But that's really only one half of that interaction. So the soil microbiome influences the soil environment, but changes to the soil also affect the microbiome. So for example, if pH drops, that can promote the growth of fungi over bacteria, or kind of a better example is if a fire goes through a grassland, all of a sudden there's a whole heap of carbon that's become available in the soil, and that's going to change how the microbes are interacting and utilizing those resources. So, just want to point out and demonstrate that the microbiome, the soil environment, and the above ground vegetation are all really interlinked. And I don't need to give you guys too much of an introduction or background on this. But just wanted to point out that the ecosystem that my study took place within was the natural temperate grasslands of the Victorian volcanic plains. They're critically endangered, with less than 1% of that ecosystem remaining, and what remains of it today is often found in a really degraded state and is under threat from further degradation and loss. So I wanted to give you all of that background to kind of explain the reasoning behind my questions. So we already have a state and transition model for plains grassland, which gives us insight into how disturbances have shaped above ground vegetation. And we know that disturbances can have a significant effect on the soil's ecosystem and the microbiome. So I wanted to find out if changes in the vegetation and soil conditions associated with disturbance altered bacterial and fungal communities, and whether a vegetation-based state and transition model could be applied to soil microbial communities. So does disturbance affect the soil microbial communities, and does it affect it in the same ways as it's affecting change above ground? And so here's how I set out to do it. I looked at five condition states from a state and transition model, the first of which was the herb rich, and that is meant to represent kind of the closest thing to a remnant grassland. So these areas were dominated by kangaroo grass and support high diversity of native herbs. So things like chocolate lilies, lemon beauty heads. And the next is the kangaroo grass dominated grasslands. And this state is similar to herb rich, but it's lost its native herb diversity through high grazing pressure. And the next state that I looked at was the spear and wallaby grass dominated grasslands. And this occurs through sustained high grazing pressure. So because livestock generally prefer to graze kangaroo grass, if grazing pressure is high over a long period of time, that gives the opportunity for spear and wallaby grasses to start outcompeting the kangaroo grass. So that's how that state is kind of occurring. And next is our nutrified grassland. This occurs through some more direct intervention, so fertilizer application, introducing pasture grasses, weed invasion, and really high sustained grazing pressure. And finally, the fifth state that I looked at was a serrated tussock dominated grassland. This wasn't actually included in the original state and transition model, so I decided to include it because when I went to look for sites for my study, it was just a massive landscape feature. I just was seeing serrated tussock dominated areas everywhere. So we decided it was probably important to understand the mechanisms behind that. And so for the sake of my model, I've assumed that serrated tussock dominated grasslands are occurring mainly through invasion to the spear grass and the nutrified state, because those are the states that tend to have a bit more bare ground available for it to be able to come in and become a monoculture. So those are my five states that I looked at. And for each state, I had 12 replicates, totaling 60 sites all up. And each site was a 20 by 20 meter area where I went out and recorded the above ground vegetation and also took 12 soil samples from across the site. This was my soil sampling layout here. And I mixed all of those together and used that to look at the soil's physiochemical properties. So that's things like organic carbon, phosphorus, pH. And I also extracted DNA from the soil samples to look at the fungi and bacteria living in it. And this here is my study area. Those of you with a keen eye might recognize that this is the Western Grasslands Reserve. That was the main focus of my study. And for those of you who don't know, the Western Grasslands Reserve is a proposed grassland conservation area. It's currently still being acquired by the state government, but once it's all acquired and managed by parks, it'll be the largest continuous area of plains grassland in the state, which will be really cool, but right now it's still mainly privately held and managed for grazing. And you can see I also looked at a few other areas. So I went out to Truganina Cemetery and Bush's Paddock to get a couple of extra kind of high quality sites. So using the soil physiochem data, I wanted to see if the soil environment was significantly different between my states. So I ran a perman over on 6 of the key variables that I was interested in, which I've got listed up there. And that showed that all of the states were significantly different from one another, except for the herb rich and the femida state, kangaroo grass state, sorry. And to have an idea of what those differences were, I'll show you a couple of plots. So this here is showing how organic carbon varied between states. and a quick recap on box plots, but the middle line there is the mean. This box is where 50% of the data lies, and then the tail ends are where the other 50% lie. And the weird blobs are kind of showing you the distribution or the spread of the data. So you can see going from our herb rich to our more nutrified sites that mean organic carbon was generally increasing, and the same was seen for the most part for phosphorus and for nitrogen, where the variability and the mean phosphorus was lower in our herb rich sites and higher in the nutrified. So now into some of the microbial data. I said at the start that the goal of my study was to see if the soil microbiome was distinct between our states. Sorry, I should say I only looked at bacteria and fungi just because of time constraints. And I found that for fungi that there was no differences in the community composition. As for bacteria, there were some differences. So the serrated tussock state had a significantly different bacterial composition when compared to the nutrified and the kangaroo grass state, and the nutrified state was significantly different from the speargrass state. So there's some differences happening, but because those weren't uniform across all the states, it's really hard to pick apart what's going on or why these states are different from one another. So I can't really speak to what these differences are or why they're happening, but hopefully that's something I can find out going forward with my master's. But something that has a little bit more juice to it, I also looked at species richness, not just composition. So species richness is just the number of unique species that I found at each site. So these large circles are showing you the estimated means, and the tail ends are where 95% of the data lies. And the smaller dots are showing you just the distribution of the raw data. So you can see overall that there was a trend of wherever there was a decrease in fungal species richness, this seemed to coincide with an increase to bacterial species richness, which is pretty cool. And it's something that's been seen in other people's work too, where fungi and bacteria have this kind of antagonistic growth, where if one's proliferating, the other one is not. Um, and so one of the cool trends that I wanted to point out here was that it seems that the transition from herb rich to feminine grasslands is occurring alongside a decrease in fungi richness and an increase to bacterial richness. And that might be persisting into the speargrass dominated states. Um, because there was no difference in the soil's chemical composition between the herb rich and the feminine states, that kind of has me thinking that maybe that initial change in richness is more driven by changes to vegetation structure. So maybe the lower native perennial herb diversity or other impacts of grazing, like lower biomass accumulation, compaction, differences in the types of organic matter being deposited. I can't say for certain, but it's pretty interesting and it's something I kind of want to uncover more going forward. And the other cool trend that I wanted to point out was that going from the speargrass and the nutrified state to the serrated tussock state, we saw an increase in fungal species richness and a decrease in bacterial species richness. So again, I haven't done all of the stats required to like confidently say what's driving that change, but it's pretty interesting and it's kind of left me with a lot more questions. So like we know that serrated tussock is a massive threat. Is it something that could also be altering the fungal microbiome and the bacterial microbiome? Is it favoring the growth of fungi over bacteria? And is that something that's occurring like directly through something it's like root exudates, or could it be just a higher biomass accumulation in serrated tussock dominated areas? I'm not sure, but I think that answering that question could have some really good implications going forward. So back to my key findings. My main question was, can a vegetation-based state and transition model be applied to soil microbial communities? And pretty much no was my answer. The vegetation-based state and transition model didn't describe, or like the microbial communities weren't well captured within those states. However, we did find some differences in microbial species richness that appeared to be linked to disturbance and vegetation change. And that's something that I'm hoping to dig into more in my master's research, where I'll be hoping to investigate the relationship between vegetation, soil physiochem, and microbial communities a bit more to see if the microbiome changes at different thresholds to the above ground vegetation. So if we know that disturbance is changing what's happening above ground, at a certain rate, maybe it is affecting below ground, but just faster or slower. Yeah. And I have a lot of people to thank for helping me with my research, especially my supervisors, Wyndham City Council, and Vanessa and Sarah from Wyndham City Council for their financial support of my project. Steve Sinclair for helping me get access to long-term monitoring data, Rupert McDonald for his help in the field with collecting my soil samples at Truganina Cemetery, and I can't quite get through all of the other names, but yeah, thank you everyone who helped. And if you have any questions, happy to answer them.
That was fantastic. Okay, so a lot to get into there. That's good. Good, great, remarkable. Where David Unwin has a question regarding the bacterial fungal species richness. Was the moisture level in the soil considered? Do you think that would have an effect on these ratios?
Um, that's a good question, and probably I didn't have soil moisture as one of the soil variables that I was measuring, but I have a few other things that could be sort of a proxy for it. Yeah, I'm not sure, sorry. That's not the best answer, but I'm not entirely sure. Yeah.
It's good to acknowledge one's limits. It's Jerry Ho has a question, might the fungal diversity in herb-rich grassland be driven by buscular mycorrhizal fungi diversity rather than the consequence of antagonism with bacteria?
Yeah, totally. I think that's kind of my pet theory at the moment is that it's like higher above ground herb species richness is maybe meaning that there's more types of mycorrhizal fungi. But I think where it comes in with the antagonistic relationship is that if those fungal species are dropping out because their partners aren't present above ground, then that is what would be allowing the bacteria to kind of increase in their richness, if that makes sense.
Hmm, indeed. Can you tell us a bit more about the fungi bacteria fight? Like, do we know what causes that at all? Uh,
I'm honestly not entirely sure what causes it. It's just I should know because I talk about it a lot. But yeah, I'm not sure what causes it. It's just something that has been seen in a lot of people's work where I think they're probably both competing for similar resources. Yeah.
okay, and do they perform the same? They don't perform the same function in the soil. Like, do you've got like mycorrhizal fungi help nutrient uptake and so on? Does bacteria help nutrient uptake and so on? It's just in a different pathway, or
Yeah, it can do, especially like um bacteria can also have symbiotic relationships with plants, which is a little bit misleading of me with my last answer. But I think that they tend to form relationships more with Fabaceae. But yeah, I guess more like broadly speaking, I think fungi are generally helping with uptake more so, but the bacteria are helping with like the breaking down of material into nutrients that can then be
Hmm,
cycled with the
right?
fungi. But I could, yeah,
Okay,
not
it,
that I know
very
for
very
sure.
complicated. Bonnie Gilman has a question. Were any of the landowners hoping to increase the quality of their sites, the biodiversity, to be better custodians?
Yes, some of them were actively um doing that. So I think Vanessa and Sarah at Wyndham City Council, they already work with quite a few of these private landowners to manage smaller parcels of their land that are higher quality. Um, so yeah, absolutely some of them were, especially because within that Western Grasslands Reserve area, like a lot of them know that it's going to be acquired anyway. And I'm definitely not saying everyone's on board and keen as to get a head start on like increasing the conservation value, but definitely some of them. Yeah.
Brilliant. Okay. It's fantastic. Other questions, folks? You can think about them as other people go along. All good. Well, let's wind up for the moment, Maggie, and we'll come back. There'll be more, no doubt, by the time we've got to the end. Thank you. It was brilliant.
Thank you.
Susan. Susan. So not just Susan. Susan is a master's researcher at Trobe, studying the demography and reproductive ecology of Ptilotus macrocephalus, featherheads, in the fragmented grasslands of the DDP. Dr. John Morgan and Dr. Susan, how do you pronounce it? Hobie, are her supervisors, and she is investigating whether small and isolated populations experience allele effects, which are reductions in fitness at low population sizes or densities. And she's also exploring the interactions between featherheads and diverse invertebrate communities, including exotic species, no doubt. And she did a BSc in plant science at the University of Tasmania. Susan, over to you.
Thanks, Adrian. So pleased to be here. Hi, everyone. Nice to meet you, and really great to see so many familiar faces in the audience as well. So yeah, I'll get started. This evening, I'll be telling you about my research, which started as an honors project in mid-2024, and I've extended into a master's degree through La Trobe's honors to master's pathway. I hope to submit my thesis and publish some papers from this work in the coming months. In my study, I used Ptilotus macrocephalus as a model to explore the challenges faced by small and fragmented populations of native grassland plants. But before we get into it, I'd like to start by acknowledging the traditional owners of the lands on which this research was conducted. I recognize their deep and ongoing connection to country and pay my respects to elders past, present, and emerging. To set the scene, I began my research by revisiting historical records of featherheads to see which populations were still persisting. What I found was quite confronting. Of the 40 populations I revisited, 12 had disappeared completely. Another 7 were hanging on as tiny remnants with fewer than 5 plants remaining. In other words, nearly half the populations I surveyed were either already gone or reduced to just a handful of individuals. Discovering that so many populations had disappeared or were surviving only as tiny remnants immediately highlighted the need to understand what was driving these declines and whether further losses could be prevented. For the sites where it's still present, we generally don't have historical population counts, so I can't say whether those populations are shrinking, stable, or recovering. All I know is that at least some featherheads are still persisting there. That raises an uncomfortable possibility. Some of these surviving populations may not be secure at all, but instead be on a path toward extinction. One of the challenges here is that individuals of this once common species are extremely long-lived. Featherheads invest heavily in below-ground growth to survive disturbance and periods of drought. In fact, this taproot was previously dated at around 300 years old. Yeah. This longevity can conceal a slow demographic decline, with plants persisting long after recruitment of new seedlings has slowed or stopped. Anyways, my work takes place across landscapes most of you would know well. The Victorian volcanic plain, where extensive habitat loss and fragmentation have left many native grassland species confined to small and isolated remnants. One of those threatened by the loss and degradation of this important habitat is this very iconic perennial forb endemic to the grasslands of southeastern Australia. Ptilotus is a diverse and widespread Australian genus adapted to dry environments, making it a useful system for examining how even resilient species respond to fragmentation and habitat loss in temperate grasslands. Unfortunately, reports of low seed set and further losses to an unidentified predispersal seed predator have raised concerns about whether populations of featherheads can actually sustain themselves. When habitats become fragmented, populations tend to become smaller and more isolated. In many species, this increases the risk of recruitment failure, where too few new individuals are entering the population to replace those that are lost. This creates what we call a demographic bottleneck, where population growth slows, stalls, or becomes negative, even if adult plants are still present. The processes that turn small populations into demographic bottlenecks are known as allele effects. These occur when survival or reproduction decline at low population size or density. For example, if plants receive fewer pollinator visits or experience higher rates of seed loss. In such a fragmented landscape as the VVP, the observations of low seed set and high predispersal seed predation raised an important question. Are featherheads experiencing an allele effect? If so, I expected to find that recruitment, rates of floral visitation by invertebrates, seed set, and early seedling growth and survival would be diminished in small or sparse populations relative to large or dense ones. I also thought I might find small populations suffering from higher rates of seed predation because where seed supply is low, predators will need to eat a large proportion of what's available to be satiated. Beyond these broader population effects, I also examined whether the local flowering neighborhood around a plant might influence seed set. Depending on how closely plants are clustered with nearby conspecifics, they may experience differences in pollinator visitation. Here, some plants are closely surrounded by neighbors while others are relatively isolated, and this may affect pollination and ultimately seed set. To test this, I measured distances to nearby flowering neighbors in the 4 cardinal directions around each focal plant and used those measurements to calculate proximity-weighted density scores. The processes I wanted to look at are all interconnected, but seedling recruitment really sits at the heart of population persistence. So I began work at my 15 study sites by looking for evidence of new individuals entering populations in the field. I assessed recruitment by measuring each plant's basal diameter as a rough proxy for age, working on the assumption that smaller plants tend to be younger. Populations with regular recruitment tend to show a characteristic pattern in their size distribution. Most individuals fall within the smallest size classes, with numbers gradually declining as diameter increases. This is known as a reverse J distribution, like in the example shown here from one of my sites. And here are the size class distributions from all of my study sites, arranged by population size from smallest to largest. What you'll notice is that many of the larger populations, particularly those with over 120 plants, show strong recruitment signals with that characteristic reverse J distribution, and most of the individuals in the smallest size classes. The fact that most of the smaller populations don't have this hints at a possible threshold population size below which successful recruitment becomes much more limited, potentially creating a demographic bottleneck. So recruitment appears to be weaker in small populations. That brings us to the key issue: what's causing it? If recruitment is constrained in small populations, there must be processes underpinning that pattern. One of the most common in plants is pollen limitation. So next I assessed whether visitation by insects differed across population sizes and densities. To explore this, I recorded floral visitation across 6 sites using video cameras, also capturing specimens to be identified back in the lab. In total, I observed over 80 invertebrate taxa visiting the flowers, spanning multiple insect orders, including those shown here, pointing to a generalist pollination system rather than reliance on a single group. Interestingly, visitation showed only a weak trend toward higher rates in denser populations, and this didn't necessarily translate into higher seed set. So while pollination may not fully explain recruitment patterns, it became clear that featherheads play an important ecological role, supporting diverse invertebrate communities across the VVP. even looking beyond pollination. Many taxa use the inflorescences as habitat or foraging ground, including spiders as predators and thrips, which live, breed, and feed on the flowers, and may also contribute to pollination, given they were seen carrying pollen. Taken together, this suggests featherheads may function as a keystone floral resource in the fragmented separate grasslands they occupy. But visitation alone doesn't guarantee reproduction. So next, I investigated whether the production of filled seeds was affected by population size or density, and the extent to which developing seeds were lost to predation. To measure seed production, I dissected every floret from each flower head and scored the contents on a 5-point scale, ranging from clearly empty to clearly well-developed seeds. This allowed me to calculate seed set as the proportion of florets that successfully produced a seed. Most seeds were straightforward to classify, but those in the middle, what I called rank 3 seeds, were more ambiguous. To determine whether these were actually viable, I used X-rays and a small germination trial in a growth cabinet. And as nearly 2/3 germinated within 3 weeks, I treated rank 3 seeds as filled seeds in all subsequent analyses. As I expected, seed set turned out to be relatively low, about 14% of florets on average. Some populations set seed quite well, but others performed more poorly. However, this variation wasn't clearly explained by population size or density. Actually, local flowering density turned out to be a much stronger predictor of seed set than overall population size or density. Seed set was significantly better in plants within higher density patches of conspecifics, suggesting that more isolated individuals are probably receiving fewer visits from pollinators, or those pollinators aren't carrying compatible pollen. In terms of seed lost to predation, I found that, as predicted, the proportion of eaten seeds declined significantly with increasing population size. These bore holes in the base of the florets and frequent observations of caterpillars point to moth larvae as the likely culprits. Also, I suspect it's probably pyralid moths that are responsible from a little matching exercise I did between plants with eaten seeds and those on which I'd spotted moths or their caterpillars. But the story doesn't end at seed production either. Even where seeds are produced and escape predation, persistence ultimately depends on what happens after germination. To examine this next stage of the life cycle, I conducted a glasshouse experiment comparing germination, early growth, and survival using seeds sourced from 7 populations that differed in size and density. This was done under uniform conditions, allowing me to remove the effects of environment, meaning that any differences in germination or seedling performance reflect source population context rather than local growing conditions. The idea here is that conditions experienced by parent plants can influence how their offspring perform, so-called maternal or carryover effects, and the glasshouse trial allows us to test for these directly. Given the field patterns, we might have expected seeds from smaller populations to perform poorly, but that wasn't the case. Germination was high, almost 95%, and it didn't depend on source population size or density. Germination rates, time to true leaf emergence, and early survival also showed no clear pattern. The differences emerged later in growth trajectories. Seedlings from small and low density populations actually produced leaves more rapidly than those from larger or denser populations, but without any pronounced difference in overall seedling size. Importantly, this was the only consistent difference detected, suggesting that fragmentation is not reducing the intrinsic quality of seeds or seedlings during these earliest life stages. Overall, the patterns I observed only partially matched what we would expect under a strong allele effect. So, what do we take from this? Well, firstly, the high rates of local extinctions I identified highlight the need for reintroductions to support connectivity, pollinator movement, and gene flow. Stronger recruitment signals and lower seed predation rates in larger populations indicate that augmenting small populations through plantings could improve both seed survival and recruitment. Seed set was higher where flowering plants were clustered, regardless of overall population size or density, suggesting value in enhancing flowering density within patches. And finally, if recruitment failure in the field isn't being driven by poor seed quality, then even small populations still have important conservation and restoration value. This is encouraging because it suggests these populations may still be able to recover if the conditions needed for recruitment can be improved. It also suggests that the key barriers to recruitment are occurring in the field. Which brings me to the importance of disturbance regimes. Without periodic burning, dense grass cover can suppress seedling establishment, meaning carefully timed autumn burns may be essential for long-term persistence. Throughout this project, I kept asking myself what it would take to see fields of featherheads like this again. After my lab and glasshouse work was finished, I had surplus seeds and plants left over from the study. Rather than discard viable material, I redistributed it into active grassland restoration efforts by working with land managers and restoration practitioners. A bunch went to Melton City Council for planting at Mount Cottrell Rec Reserve, while others are supporting restoration at Cherry Creek Youth Justice Precinct in Little River. Some seeds were returned to Simon Jolly to help bolster recruitment of his small population, and I've recently sent 42 plants, many of which had already flowered, to Nangak Tamboree Wildlife Sanctuary, where they will be used for seed production to support future restoration efforts. Finally, to extend my glasshouse findings into the field, I've retained 245 plants for a restoration trial, which is currently planned for Illaborook Grassland Flora Reserve. These are small steps, but they're tangible ways to move from documenting decline to actively supporting recovery. That brings me to the end of my presentation. Before I finish, though, I'd just like to express my gratitude to everyone acknowledged on this slide who has supported me throughout this project. And also thank you very much for your time and attention. I'd be happy to take any questions. Oops.
Babo, fantastic, thank you. Brought backup slides. Okay, we'll ask you about them in a sec. There's one comment in the chat asking if people could possibly be taking seed. Is that could that be impacting what's going on out there at all?
Yeah, I suppose it's a possibility.
Okay. We'll have to have to put out cameras and try and catch them at it maybe.
Yeah, I suspect the bigger issue is the management of a lot of these sites where grass is overcrowding the featherheads and not allowing them to recruit.
Right.
Yeah.
Did you do actual did you measure like the amount of biomass around and so on? Is this something you really explored, or is that more like a gut feeling kind of observation?
It's more of a gut feeling observation. I did try and sort of estimate the percentage of grass cover versus bare ground versus herb cover at each of the sites. But I'm not seeing any like clear correlation between, for example, bare ground and recruitment or seed set or anything like that. Yeah, because I'm sure there are other factors that sort of play into whether they're able to successfully recruit new seedlings. Yeah.
Mm-hmm. Yeah, and what's your what? Oh no, I know the answer to that already. Sorry, I don't have to ask that. More questions. Lee has asked if there's perhaps preferential grazing from sheep and goats. So do Ptilotus get grazed by particular things in preference to other, or do certain species graze them more than others?
Well, I don't think there were any sheep or goats at the sites where I was doing my study. Some might have kangaroos. I don't know if as far as I know, they don't eat featherheads. I've heard that they do get nibbled on by slugs, like the leaves will. And I've seen many of the the floral heads get chopped off, which I think is done by cockatoos or corellas.
Okay. Uh, And the slugs, they're all introduced. Oh, John Del Pratt's got a question. Very good. John.
Hi, Sue. Thank you very much. That was wonderful.
Thanks, John.
Interested that you counted florets within an inflorescence. Did you just do the one? How many did you do? How many
Yeah,
different florets?
I didn't mention that. I collected 15, a random sample of 15 heads from different plants around each population.
Did you find what I found in the past, that a lot of the filled seed seems to be at the base of the inflorescence, or is that not a pattern that you observed?
Yeah, I did notice that. Yes.
Hmm. Something to think about sometime.
Hmm.
And your 14% was filled seeds, regardless of whether they've been predated or not. Was it? Or
Filled seeds.
you said 14%.
Yeah, so that's excluding the predated ones.
Excluding them, because that seems fairly high to me, actually, having done the same thing. Yeah, you can do a lot of searching without finding in those heads, but yeah, that's good. Okay.
Yeah, it was quite a encouraging, I think, finding because we could have expected to find less. Maybe about a quarter or so, I can't remember the exact number, of the heads that were collected had no seed set whatsoever.
Yeah.
So there was definitely a lot of variation as well.
And I have had one season in my time where there was enormous seed set, so it does appear to be again, one of those species that has variable seed set depending on conditions. But
Yeah,
yeah,
absolutely. Yeah. So it'd be interesting to repeat this in a different year, with perhaps
yeah.
with more rainfall or something to see how much it differs.
Thank you. Terrific work. Thank you very much.
Thanks, John.
So in terms of like growing from seed and so on, is it an easy process to do? With those long roots, is that something that you need extra long containers for, or
Yeah,
um,
well, they are really easy to germinate. I didn't
mm,
didn't need any pre-treatment, didn't need to sterilize the seeds or anything. Just put them in native potting mix, in forestry tubes. So they're not, they're not very long. They're like 12 centimeters long,
mm,
just to get them started. And then to get them to flower, I did transfer them into bigger pots that are taller, to give them room to grow. And yeah, just gave them some flower and fruit fertilizer, and off they went. So yeah, actually, yeah, they grew really quickly and really happily that way.
okay. Oh, good.
Yeah.
Vanessa.
Yeah, thanks very much. That was a fascinating talk because I was really keen on this because I've seen those plants come and go and go and go and go, unfortunately, over the years. Question, given the size of the taproot, to what extent do you reckon you can get any recovery once they're not being visible for a year or two? Do you think they actually have any degree of dormancy, or they're just basically, once they don't pop up leaves and flowers, they're gone?
Yeah, I couldn't really answer that. I don't know the answer to that, but I like to think that they can hang on below ground for quite a long time and then pop back up when conditions improve. But yeah, I don't know for sure. So
Because that leads to the question is, would you be prioritising supplementing existing populations, or would you even have a stab of chucking in some more plants in places where they used to exist, such as, mega secure sites like Evan Street, Sunbury?
yeah, absolutely. I think we need to do all of those things.
Fair enough. Thank you.
A couple more questions in the chat. Climate change, is that influencing your findings, do you think?
I imagine that in low rainfall years, they probably don't set seed as well, don't produce leaves as well, just generally don't grow as well. So yeah, definitely climate change is making things warmer and drier, and that could be exacerbating their plight for sure.
And from Frank, how significant is the loss of traditional cold burns to the survival of these species, do you think? We've sort of talked about that, but the
Yeah,
cold burn side of things?
yeah, massively. I think definitely those sites that get overgrown with grass or weeds and where seeds just don't have an opportunity to germinate and grow into adult plants. Those are the ones that are faring the worst, like just from my own observations. So I think burning regimes, like I mentioned, are really important for maintaining those sites and maintaining successful recruitment throughout the seasons.
And seed viability. Do we have an idea how long, this is from Robin Tyson, how long you think the seeds might remain viable for?
Seed longevity,
Yeah.
in the field, I think they have a pretty transient seed bank. They're not something that retains like a what is it called? A persistent seed bank. So they do rely on seed dispersal quite frequently to maintain recruitment numbers. But yeah, that'd be interesting to test. I don't know if maybe the Botanic Gardens or something has already done some testing on seed longevity, but I've still got some seeds that I've decided to hold on to so that I can do another germination trial in, I don't know how long. I have to decide with my supervisors whether we wait 12 months or something and see, yeah, whether the viability of those seeds decreases over that amount of time. It'll be interesting to see.
We're zooming through the time, so we better get on to Griffin. We're going to have a late night tonight, and that's absolutely fantastic because these talks are brill. Thank you very much, Susan.
No
Excellent.
worries. Thank you.
And we'll yes, there will be more questions at the end. Griffin, on to you. Griffin Clements has recently finished his honours studying button wrinklewort, and he looked into its fire survival and regeneration failure. He runs invasive replace, which is a small indigenous nursery to grow plants to regenerate urban green spaces with indigenous flora and to remove invasive plants. So, Griffin, thank you. Welcome. Do take over. Griffin, you might be on mute. I'm not sure. It's
Thanks, Adrian. That's better.
it is better.
Perfect. So I'm Griffin. Last year I did my honours studying the button wrinklewort, or the Rutidosis leptorrhynchoides, where I looked at its early life history vulnerability to cilian herbivory, and I also looked at its fire survival throughout the year. Here you can see some of the seedlings that I grew out in the field, and they're just little tiny babies in this photo. So the button wrinklewort is an endangered grassland daisy. It's endemic to southeastern Australia, where it was found throughout the Victorian Volcanic Plains grassland, as well as the Gippsland Plains, and up into Canberra and New South Wales. It lives for up to 15 years, so it's quite a short-lived species compared to some species like Ptilotus, and that means that if it's not regenerating, then we don't have very long to fix that before we start losing populations. We know that it will not persist long term without regular fire, and we know that it suffered a massive decline since colonization. It was once a common and widespread species. In fact, it was so common that it was a co-dominant species with kangaroo grass. It suffered a massive decline, predominantly due to intensive sheep grazing, as this species is particularly vulnerable to intensive grazing. It's also had a decline due to agriculture, urban development, and particularly changes to management practices of grasslands, specifically changes in fire practices. Because of this, it's now restricted to just 24 populations throughout Australia. And on top of all of that, it's suffering a regeneration failure. That means that despite producing viable seeds, none of these seeds are actually making it to adulthood. In my study, I broke up the plants into a few different phenological stages. You can see in the first photo the basal rosette stage, where leaves grow from its central crown, and this is its winter wheat regrowth stage. After that, it would enter its elongation stage, where it grew flowering stems, and then it would enter its flowering stage, and finally it would enter its dry stage, where it would become dormant for the year. So there are no native slugs in Victoria's grasslands. There are, however, many invasive slug species now present. Slugs are known to be a problematic seedling herbivore, as they eat at ground level and they love the taste of juicy little seedlings. And we also know that because there's no native slugs, our plants don't have natural protections from slug herbivory. Rutidosis has been found to be highly palatable to invasive slugs, and this all led us to believe that slugs may be consuming Rutidosis seedlings, contributing to their regeneration failure. As we all know, fire plays a key role in the Victorian volcanic plains grassland. It clears away overcrowding dominant grasses, allows the rejuvenation of plants, and creates a clear seedbed for regeneration. Rutidosis specifically relies on regular fire, and as I mentioned before, without regular fire, it will fail to persist in any grasslands. In fact, at the tetraploid populations, up to like near Rokewood, a lack of fire recently led to a massive decline of those populations, where they're only down to a few individuals now, sadly. We don't yet understand the best time of year or phenological stage to burn Rutidosis to minimize its mortality, so that's what I set out to discover. For my regeneration failure experiment, I wanted to estimate the invertebrate composition of the Truganina cemetery grassland, and to do that, I spread out 10 pot trays throughout the grassland randomly, and then each week I went out and I checked those trays and recorded all invertebrates that were found. I only found a single slug and a single snail the entire time. The most commonly recorded species was the Portuguese millipede. Isopods were also found frequently, and I also found ants, beetles, spiders, and centipedes. So to try and understand if slugs were eating the seedlings, I undertook a slug exclusion experiment. I did this before I realized there'd be no slugs, as I did the faunal composition experiment at the same time as the slug exclusion experiment. But what I did was I compared germination. What I did was I planted out 10 seeds in each of 60 sites throughout the Truganina cemetery. In half of those sites, I dug up the ground beforehand to compare just germination in disturbed and undisturbed soils. I then compared the survival of the seedlings in 3 different types of protection. I protected the seedlings with copper tape because that creates a physical barrier to slugs, as it will electrocute them if they try and touch the copper barrier with the copper. I also used methylhydride slug bait, which will just poison the slugs, and then I also had to control unprotected sites. Every week I went out and I checked the seedlings, and I saw how many had germinated, and I saw how well they were surviving. I marked each one with a little toothpick, so I'd know that I'd already recorded that one. And I spent most of the time getting swooped by a magpie, so that was a lot of fun. I found that germination was particularly high in disturbed soils. You can see in this graph here that smaller bar represents undisturbed soil, and we only had 1% germination across all the seeds planted. We had 4% germination across the seeds planted in disturbed soils, which is a significantly higher amount. However, germination was particularly low in the slug bait protected plots. I only had 2 plants germinate in the slug bait plots, while in the unprotected and the copper tape protected sites, germination was pretty consistent. So I undertook a Kaplan-Meier survival graph to compare the survival rates across the 3 treatment groups. As you can see, this graph shows how the survival probability changes throughout time, across the 3 different treatments. You can see that all 3 treatments reduced at a very similar rate throughout the whole time, suggesting that there's no difference in the probability of survival between the different treatments, which is not surprising as there was no slugs to actually eat the seedlings. So to put this all in context, soil disturbance had a clear impact on germination rates due to disturbed soil being less dense and that increasing seed-soil contact. Mollusk abundance was exceptionally low. This is likely due to it being such a dry year last year, and also the fact that this was undertaken recently after a fire in the grassland. Non-native slugs are widespread in the Victorian Volcanic Plains grasslands, and their slug numbers and their impact are likely to be greater in a wet year. There was no difference in the survival probability between protected and unprotected seedlings. This suggests that slugs did not play a role in the regeneration failure of Rutidosis, at least this year. Millipedes and isopods are also invasive seedling herbivores, so they may be playing a role in the regeneration failure. Slug bait likely had a negative impact on germination due to the fact that it has a toxic effect on plants. So I also undertook a 5 survival experiment. So every 2 weeks, I grabbed 10 plants that I'd been growing in the greenhouses, as you can see behind me, which I'd up-potted into these larger pots, and I counted every single leaf on each plant to measure their size, and then I burnt 10 plants using a weed burner that I had set up on a stand, so it was always coming from the exact same position, and I burnt each plant for 40 seconds. I also defoliated 10 different plants using a pair of scissors where I'd cut them off just at the base of the soil. I then compared the survival across their different phenological states and throughout time by seeing which ones had resprouted after a month. So these graphs show the phenological states of the plants throughout the experiment. As you can see, they start off in their basal rosette stage, and then they slowly enter their elongation stage. Flowering grows quite consistently throughout the experiment, while they enter their dry phase right at the end, because we had a few weeks of extremely dry and hot weather that forced many of the plants into dormancy. So rhododosis was found to be susceptible to fire throughout the test, with mortality ranging between 80 and 10%, depending on each experiment time. Mortality was highest at the start of the experiment, which was in May, and it was lowest around November. It did increase again near the end of the experiment, and I'm still looking into why that may have been. They survived defoliation better the entire time that we were experimenting, as you can see in the blue line. And they followed a similar trend where they died off most at the start, survived the best around November, and then decreased their survival rates again. I also looked into if the plants would reflower after the burn or defoliation treatment. As you can see, plants burnt very early on did not reflower well, and plants burnt very late did not reflower well either. However, plants burnt around November did manage to reflower in that same year, in that same flowering season, suggesting that they would be able to reflower in the wild. Plants that were defoliated consistently managed to reflower after regrowing from their base. I found that size did not determine the fire survival rates in the burnt plants. As you can see in this first graph, there's a pretty consistent probability of resprouting across the different sizes. But in the defoliated plants, I found that larger plants were more likely to survive the defoliation. You can see that between 100 and 200 plants, leaves, the plants go from having a low chance of survival to quite a high probability of survival. So to put this all in context, the seasonality of fire has substantial impacts on its intensity and how it affects plants through their interactions with phenology. Like other plants, Rutidosis was more susceptible to fire than to defoliation. Rutidosis was very susceptible to fire during its winter regrowth stage or basal rosette stage, as well as during its early spring elongation stage, becoming more resilient to fire around November. This suggests that a winter or spring fire are likely to cause high mortality in the wild. So our overall findings were that germination was exceptionally low in the field, with only around 4% of seeds actually germinating. There's clearly something going on there that's not helping this plant grow. We do know that in general, grassland germination and plant survival is quite low, but we would really be expecting to find that this plant is managing to regenerate, but we're just not in the remnant populations. Soil disturbance drastically improved germination. Slugs were not a driver of the regeneration failure this year, but they may be in a wet year. Slugs may still be a problem in a wetter year, and Rutidosis is vulnerable to fire in winter and spring. Early application of fire or defoliation increases reflowering ability. Some future research I'd love to see happen on the Rutidosis is I'd love to see this slug exclusion test redone in a wetter year to see, does a wetter year result in greater germination, and does the wetter year increase slug numbers and therefore their impact? I'd also love to see someone test if millipedes or isopods are consuming Rutidosis seedlings, and this could be done through a laboratory food preference test, as has already been done with slugs, as well as in-field exclusion tests. I'd love to see a continuation of the fire experiment throughout the rest of the year. Unfortunately, I didn't have enough plants to go for the whole year, and see when Rutidosis is less vulnerable to fire. I'd like to acknowledge John and Steve, Dr. John Morgan and Steve Sinclair for being my supervisors, Maggie Logan for helping me set up my field experiment, Rupert McDonald and the Bunurong Land Council for allowing me to conduct this experiment in Truganina Cemetery, and the Nangat Tamboree Wildlife Sanctuary for providing me with the plants. Thank you. Any questions?
Brilliant. Thank you, Griffin. That was very, very interesting and good to see a bit of failed science, for want of a better word, as well. It's negative results are just as interesting as positive results, aren't they? So burn in November. That's a important point.
Yep.
And yeah, that low germination. So do you have any idea what's going on with the low germination? That's all.
I also did a grow cabinet germination, and I got a higher germination rate, around 20%, which is still pretty low, but it's much better than it was in the field. So clearly the dry year would have had an impact. And I just think that a lot of the seeds didn't have a chance to get into the soil when it was undisturbed because the soil was quite compact and hard. So I think that plays a huge role. But I'm not sure why we had quite such low germination rates. It was shockingly low.
Yeah, amazing. John Del Pratt, you have a question.
Thanks, Griffin. That's terrific. I may have missed it. What was the latest burn that you did?
So I went until the very end, the start of this year.
Sorry, what month? Oh, you're talking about
January.
So there was more. The fire had more of an effect in January than in November.
Yeah, interestingly, I found that very surprising.
We find that Rutidosis is one of the later flowering plants in the landscape. So, but I can't quite work out why. In spite of that, I can't work out why it would have been more susceptible in January. I mean, normally you would expect the burns to be recommending autumn burns for something like that, but that doesn't appear to be supported by what you found.
Yeah, part of the problem with my experiment was around the very end of it, where we had those plants go into that dry stage and went into their dormant stage. Some of them then resprouted, and so we're still looking into if potentially that unnatural resprout, caused a high mortality. Well, I'm still investigating that part to try and publish.
I would strongly support that hypothesis.
Yeah.
Yeah. Okay. Thanks very much.
Jessica's got a question, which is given that button wrinklewort appears to germinate better in disturbed soil, do you reckon that's got the local extinction of native digging animals like bandicoots and so on is going to be a factor in its decline over
Yeah, I
time?
definitely think that there's a correlation between that happening, as they would play a role in disturbing the soil, and without that soil disturbance, the seeds just don't have a clear seed bed to go into. So I think that's definitely part of it.
Has there been any research done on that?
On the idea of seeds germinating in disturbed soil a bit more, yeah, and there has been a difference found between rabbits and our native animals in that, with our native animals actually creating better germination conditions. So there has been a bit of research, but not specifically with rhododosis.
And does that change the invertebrate communities? Have they been linked to this whole disturbance?
I'm not aware of that.
No. Okay. There was a complicated question from Jessica. It had lots of moving parts in it. Yvonne and Rito ask if you check to see if the seedlings continue to survive in the disturbed site. Yvonne would have thought that they would end up having more weed invasion.
I've been meaning to go back out and check on them recently. I'm planning to go out this week and actually have a look at them and see if any of them are still going. But I'm doubtful of how dry and hot it was this year.
Okay. Andrew Arnold asks everyone if we've or asks you no, have you visited the reserve near the Hopkins River beside the rail reserve near Dobby?
No, I haven't visited that one.
Okay.
Yeah,
I'll
that
have a look.
one is actually very close to Ararat,
Okay.
and it's quite interesting. I'm finding your comment about November burning very interesting, because a lot of the Rutidosis sites that I've looked at over probably about a 20-year period or longer are beside railway lines. And if you go back to the 1970s and 80s, the railways used to do their burning in about that time frame in November, just before the severe onset of summer conditions, so that they removed the biomass from those reserves. And that may have assisted with the preservation of some of those sites, but they're probably gradually disappearing now because the burning is not still done.
That's really interesting and good to know. I think you're absolutely right that the consistent burning played a huge role in maintaining those populations.
Yes, I would agree with that.
It's incredible. I'd never realized, Griffin, that there are only 24 sites in Australia, all of Australia, where you find button wrinklewort. It's just shocking.
From one of the most common plants that we used to be around, and now it's barely exists at all.
Yeah, that's truly amazing. And what how do you know how come they let the rokewood population collapse? So it was just because it wasn't burnt for a whole lot of years. And why didn't they? Was it a COVID thing, or
I don't know why it wasn't burnt. I just know
muddy-mindedness, or
I don't know why it wasn't burnt. I just know that the lack of burning shaded them out, didn't give them a chance to grow and survive, and we've lost a lot of plants because of that.
Yeah, it's gone from 1,000 down to dozens, hasn't it?
Yeah.
Yes.
And the need for burning was known.
Yeah.
It's not a new fact, is it? Like that's
definitely.
Yeah. Well, that's just irresponsible. That's
yeah, it's pretty sad.
Yeah, it's really bad.
Luckily, we've got a
set
seed.
Like a setting seed, I think.
What's that, Deb?
A lack of setting seeds.
Yes.
And seed bank. The seed bank must have been depauperate, I think.
Yeah, they don't have a seed bank. They've got a transient seed bank.
No, very small.
They go into the ground and germinate, or they don't germinate at all.
Yeah.
I was going to say, Rutidosis are they're literally, right, rail reserves, and they're not grazed either.
Yeah, exactly.
So lack of grazing and really good burning regimes has maintained them, but now they don't even have good burning regimes.
The only good areas at Rokewood Cemetery are actually out in the road reserve.
You're right, on the roadside.
Where it has been disturbed
Mm.
by possibly mechanical scraping and that sort of thing. If you go into, and I remember thousands of plants in there going back to the early 2000s. And, you know, as you say, the loss of the burning regime has flawed that population, unfortunately.
Yeah.
Hmm.
If you want to see some beautiful button wrinkleworts, I highly recommend the Iramu grassland. They've done a great job at reintroducing them there. And it looks stunning in spring when they're all in flower.
Also, Pioneer Park looks really good too, Griffin. Will Larson did an amazing job of reintroducing them there, and they're doing it on their own because they burn regularly. So that's another place to go check them out as well.
Awesome.
You should talk to John Morgan about his reintroduction of button wrinklewort at Dobie and also a couple of other sites, but it'd be worth talking to John about that.
Yeah.
Yes, there must be quite a lot of new populations that have been set up in the last decades, I guess.
Yeah.
Oh, well, this is going back quite a few years. The John sites that I'm talking about, yeah.
All right. There's one new comment, sorry. So Julie asks, is the best time to burn wrinkleworts May or November?
Definitely around November. May was a really bad time to burn them.
Okay, good. We'll look at 7:50 now, folks. Perhaps we should, well, hold on. Are there more questions should we just throw open the questions a little bit now too, if you have anything from for Susan or Maggie that you've been realized you need to ask? Otherwise, yes? No?
I'd just like to comment, if I may,
Sure.
Griffin, the restoration west of Wycliffe, Rutidosis was one of the more successful plants there, and is recruiting quite regularly on that site. Now, that's a very tough site, lots of gaps, but it is actually actively and has frequently actively recruits there. That's the polyploid. Just it's a large population that is recruiting on site.
That's great to hear.
And David Franklin sitting there listening to all this, and that's a lot of it's down to him.
Very
and
good.
Paul and Paul Gibson Roy. Yeah.
Bonnie.
Adrian, we deal with a lot of negative stuff with grasslands. There's a lot of sadness and there's a lot of exasperation. But tonight we've had these fabulous humans who are doing this lovely work that just fills me with joy. And I say thank you so much, you excellent young humans. And I hope that what you're doing just also enthuses others to go on with it, because it helps us. It helps us be strong and keep fighting. Thanks.
Thank you.
Thank you, Bonnie. Great words, great words. And yes, so thank you, excellent young human researchers indeed. And let's wind it up there on such a absolutely fabulous note.